This will be a review of some of the important topics in 1.1. The cell theory, which you can learn a lot more detail about in the textbook and also through the TED Ed video, which talks about how the cell theory was developed and the scientists that were involved in that. But essentially it has three parts. It states that cells are the smallest unit of life. All organisms, an organism is a living thing, so all organisms are made of one or more cells. And then finally that cells arise from pre-existing cells. You've probably been learning and reviewing that particular theory since 7th grade life science, but it's important to revisit because now you need to add some exceptions to your understanding of that. So theories are often challenged, and sometimes they change as a result of that. Some things that bring the cell theory into question are some examples of cells that don't necessarily fit the mold. We're going to discuss those here. Now, the cell theory, it didn't bring it into question enough to have it negated. However, there are some interesting exceptions. So for example, muscle cells, striated muscle, they make up a skeletal muscle. So striations refer to these, it's kind of hard to see here when you zoom in that far, but basically these little tiny lines here, they look like stripes, and those are striations. And skeletal muscle, the muscle that's attached to and moves your skeleton, is striated. Well, these muscle cells are an exception to what you would commonly think of as a cell because they are really, really long, up to 30 millimeters long. And they have multiple nuclei, sometimes as many as several hundred within, before you have like your typical outer cell membrane splitting them apart. So this extreme length and also the multiple nuclei make striated muscle cells an exception to what you would commonly think of as a cell according to the cell theory. Another example is fungi. They consist of really long structures called hyphae, and then in aseptate fungi, the hyphae also have many nuclei similar to the way that the muscle cells have many nuclei. So that's another example. And then the giant algae, these are single cells that can grow up to 100 millimeters long. So that's really large for a single cell. They still only have a single nucleus, though. Also included in 1.1 is a discussion of the basic functions or qualifications for living things. So let's look at those as they relate to paramecia, which you spend a lot of time looking at in the lab. So one of these functions of life is homeostasis. Homeostasis, the prefix homeo sounds like homo, so that's same, and then stasis is stay. So when you think of homeostasis, what conditions is an organism trying to maintain or keep the same? So for example, for us, that could be body temperature. For the paramecia, that could be a balance of the correct amount of water inside and outside of the cell. So contractile vacuoles, which are vacuoles which hold water, but they can also contract or squeeze, allowing them to push water out if the water inside the cell gets to be too much, and then they can relax in order to allow more water into the vacuole. And that helps maintain homeostasis. Metabolism, remember metabolism is all the chemical reactions occurring in an organism. So all chemical reactions. And in the case of the paramecia, this occurs in the cytoplasm. They're single-celled organisms, so they don't have specialized organs. They are likely have some lysosomes that have a high amount of, or a high functioning role in this process. Response. So living things respond to stimuli. A response example in the paramecia is the use of their cilia to propel through the water toward food. And you saw this as they move toward yeast. Reproduction, the paramecia reproduce through mitosis. So that's asexual reproduction. All living things reproduce. This is actually one of the key reasons why viruses aren't classified as living things, because they do not reproduce on their own. Our cells have to reproduce virus. Additionally, viruses aren't cells, so that's another reason why they're not considered to be alive. For nutrition, they have the food vacuoles. They take in food through the oral groove, and then that food is stored inside vacuoles and digested, and then the carbon compounds like glucose are used in cellular respiration, ultimately to generate ATP. Growth, biomass, so that term bio is life or living, and mass is the amount of space, essentially, that something takes up. So biomass is assimilated biomass, so how much space does this living thing take up? And that is used biomass, so the different biological or organic macromolecules are used to increase the size before the cell divides. Excretion, for us, this is what we do in the restroom. For paramecia, they move substances out through the cell membrane. They move waste in that way. Now, when we were talking about paramecia is a single-celled organism. But there are many organisms, like us, that are made up of multiple cells. And there are specified levels of organization in multicellular organisms, and depending on the level of complexity of the organism, that determines how many of these different levels are relevant for that organism. So cells would be the smallest unit of life. Cells make up tissues. So the example here is epithelium. The organ that this epithelial tissue is a part of is the stomach, and since it's epithelial tissue, it would be the lining of the stomach. So it would be right around in here. You're also going to have smooth muscle. That would be on the outside of that epithelial lining. The organ system that the stomach is a part of is the digestive system, and then multiple organ systems work together to make up an organism. So cells make up tissues, tissues make up organs, organs make up organ systems, and organ systems make up organisms. Multicellular organisms have specialized cells or specialized tissues, meaning not all the cells do the same thing. And as a result of this, they can develop what are called emergent properties. These properties get their names because they are indicative of the compiled work of all of the different cell and tissue types. So each individual cell would not be able to do this work. It's an emergent property that's the result of multicellular components working together. And an example is like the higher order thinking, it requires messages to be sent and exchanged between cells, between neurons specifically. Now, as these cells turn into these specialized types, that actually is called differentiation. So you start as a fertilized egg cell, a zygote, and then from that fertilized egg cell, all of the egg cells that are part of an organism come from the division of that original egg cell, yet all of those are different. And that comes from differentiation. So the early cells of an embryo, in the really early stages of development, they're stem cells. And then depending on the environment where that particular cell ends up, there will be different factors released that will ultimately cause it to become a particular cell type, like a nerve cell, a muscle cell, a red blood cell. What causes this differentiation is the differential expression of genes. So again, each cell in an organism contains the same DNA or the same genome, but not all of those genes are being expressed or made into protein. And it's what gets expressed or made into protein that ultimately determines the physical characteristics of the cell. And so that's what's shown here in this schematic. This particular gene is one that contributes to the development of a neuron. This particular gene is one that's important for epithelial cells. But all of the genes are in the nucleus of each of these cell types. It's just which ones are being expressed and which ones are not. Stem cells then are cells that are not yet fully differentiated. Zygotes, of course, are the most potent, meaning they have the most potential of any stem cell because there's been no division or differentiation at all. So it is really the epitome of a blank canvas when it comes to cell types. But a zygote isn't the only way that stem cells can be harvested. There are other types of stem cells, and they fall into these categories. The zygote would be totipotent, toti for total potent potential. You may also see this as omnipotent, like an omniscient, all-knowing, omni-all, so all-potent or totipotent can become any cell type. And again, this would be the zygote or very early blastula in an embryo. Pluripotent can become most cell types. And then finally, multipotent can only differentiate into a particular class of cell. This just shows you some of the disorders that can be treated with stem cells. Okay, so this shows you what the deal with embryonic stem cells. So you have fertilization, so that's the egg, and then that's the sperm, and then that generates a zygote. And then this is after the first division, and then after several divisions, you have what's called a blastula. And from there, a cell can be harvested, and this is a stem cell. And this is showing the early stages of development where you have an ectoderm, which is the outer layer here, a mesoderm, which is the inner layer, and then the endoderm is sort of everything in between. And the ectoderm, those cells can be harvested to develop into the brain or skin. The mesoderm can be harvested to develop into muscle, blood, bone, or cartilage. And then the endoderm, the innermost area, is the lung, gut, and liver. There are benefits and drawbacks to the use of embryonic stem cells. The benefits, there's almost unlimited differentiation potential, so they're omni or totipotent, and there's less of a chance of genetic damage via mutations because they have not been exposed to environmental mutagens. The downfalls, there is a higher risk of the cells becoming a tumor because they are in such a fast dividing stage when they are harvested. And also there's ethical issues with the fact that removing the cells from the embryo kills it. Another way to access stem cells is from umbilical cord blood. These are extracted from the umbilical cord of newborn babies. And these cells are pluripotent, meaning that they're not as potent as the embryonic stem cells, but they do have potential to develop into several different types of cells. This is showing a multipotential hematopoietic stem cell, and it's showing how it could develop into these different types. These are all different types of blood cells. So these are especially useful if there's like leukemia or some other condition affecting the blood cells. This is showing the banking of umbilical cord blood, which is actually kind of pricey. And I mentioned in class how there's an ethical issue associated with this. So it's something for you to think about with a good TOK thinking session on how you feel about whether or not that's right or wrong, because it's very expensive to bank cord blood. So that means it's only available to certain individuals, and it is able to save lives if the child that was attached to that umbilical cord or someone else in the family were to need those healthy blood cells. So anytime that there's a life-saving treatment that's only available to certain groups of individuals, I think it certainly brings to mind an ethical debate. All right, so here's some basic benefits and drawbacks for umbilical cord blood cells. They are easy to obtain and store, just removed right from the umbilical cord. And oftentimes umbilical cords they're not kept, so they're just disposed of. So it's just a good use of that material. The cells came from the patient, and so they're genetically compatible. You don't have to be concerned about that. And there is a limited quantity of those in the umbilical cord. Downfalls could be that, it is expensive. The storage is expensive, and I just talked about the ethical dilemma with that. And they cannot differentiate into as many cell types as can the embryonic stem cells. And I think this limited quantity in umbilical cord should probably be over there. All right, and then finally the third type is adult stem cells. Those are taken from an adult's own tissues. They're harder to obtain because it's difficult to tell which cells are stem cells and which are not just by looking. A lot of times you have to pay attention to how the cell is behaving, and since they're in a living person, that can be difficult. And depending on how deeply in the body the cells are embedded, that can increase the difficulty as well. They also have less growth potential. They're not able to develop into all of the different cell types. The way this works is they're isolated from the patient, and then the cells, this is kind of reverting them back. So you try to find the stem cells, isolate them from the patient, then in a petri dish, meaning in vitro. In vivo is in living things, in vitro is in test tubes or petri dishes. So then you're going to, the growth factors that help to determine what the cell will become, treat the cell with those, and then hopefully from there be able to alter the conditions of the culture, and the culture is just the cell culture. That's the environment that you have the cells growing in. So whatever the culture you're providing to these cells in this petri dish, this technique is called cell culture. So that's what that word is. So you change the conditions in the culture, providing other different factors, and hopefully have them develop into whatever type of cell it is that you are needing. Although again, this isn't totipotent. Basic benefits and drawbacks. There's less of a chance of tumor development because these cells aren't in a high dividing stage. You don't have to worry about compatibility with the patient's tissue since it comes from their body. And removal of the cells does not kill the adult, as it does in the case of the embryo. The downfalls is that they are difficult to obtain, especially from deeper tissues, and they do have less potential as far as what they can develop into. One particular disease that you need to be familiar with is Stargardt's disease. This is an inherited condition, meaning that it's congenital, so it comes from a genetic abnormality. Oh, I just noticed condition is spelled wrong. There. And it results, oh goodness, there's another typo. Results in the degeneration of the macula, and the macula is a region of concentrated photoreceptors, specifically cone cells, in the back of the eye. Photoreceptors are neurons or cells of the nervous system that function specifically in vision. Cone cells are photoreceptors that are important for focused color vision. And the macula is right at the back of the center of your eye, which is why if you want to look at something and you really need to focus on the details, you look at it straight on. And your periphery of your eye, your peripheral vision, there's more rods, which is the type of photoreceptor for vision in dim light. So not as much detail there. This particular disease affects juveniles and can result in blindness if it is not treated. And interestingly, even though this disease affects juveniles, it oftentimes, the difference in vision, the impairment, may not be apparent until later in adult life. And this is how this treatment works. In 2010, the USDA or the US FDA approved stem cell research, or stem cells, for the use in treatment of Stargardt's disease. And the way that the trials work is, and ultimately the treatment is, vitamins and proteins are used to turn stem cells into retinal cells. So again, those would be the different factors required to stimulate the cells, the stem cells, to turn into the particular type of cell that is an issue in this case. Millions of healthy cells are injected into the back of the eye under anesthetic, and this can be done in a quick 1-hour operation. And then after several weeks, sight should begin to improve as those healthy photoreceptor cells that were stem cells divide and replace the damaged regions of the macula at the back of the eye. Another important concept covered in 1.1 is surface area to volume ratio. And this is why cells are so small. So a cell's volume is all of the stuff inside, all of the space that it takes up, which is essentially filled with cytoplasm and all of the organelles and the nucleus and all of the good stuff that goes on inside of the cell. So a cell's volume determines the amount of chemical reactions that can take place. So the larger the volume, the more chemical reactions, the more metabolism, the more stuff you're going to have going on inside the cell. Now, in order for all of this to be facilitated, everything that's required has to come in through the cell membrane, and everything that is generated as waste, or for the purposes of export, not just waste, but things that need to leave the cell for productive reasons, have to leave across the cell membrane. So you've got entry and you've got exit, and the more going on inside of the cell, the higher amount of volume you have, the more need for, oh, that's not pretty, the higher the need for movement across the cell membrane. And the cell membrane comprises the surface area. So all of this area here, and of course this cell, it's not showing the membrane that would be here on top of all of this because it's showing us the contents, but you would have membrane right all the way across the top. So all on the outside you're going to have cell membrane, and that's going to be your surface area. The larger the cell gets, the more volume it has, the more metabolism it has, the more it's up to, the more it's doing, so the higher the need for movement across the cell membrane becomes. And the cell membrane is the surface area. Okay, so the surface area, the amount of area available, determines the rate at which molecules can pass through the membrane. So the more surface area, the more reactants and products, waste and necessary elements for metabolism can diffuse in and out or move across. So cells with a larger volume are going to require more, and they would thus require more surface area. This is sort of like an office building. If you have an office building and it has 1,000 employees, you're going to need more entrances and exits than an office building that has 15 employees. And the entrances and exits in this case would represent the cell membrane, or the surface area that can be penetrated for entry and exit. So when a cell grows, both the surface area and the volume increase, but not at the same rate. So this is showing an increase in the length of the side of each of these cubes, but then the volume and the amount of surface that the cubes have exposed does not increase at the same rate. So the first cube, the surface area was 6 micrometers squared. The volume is 1 micrometer cubed. But then as it gets bigger, so this is a 6:1 ratio, as it gets larger, both the surface area, as we go to 2 micrometers in length, both the surface area and the volume increase, but not at the same rate. So 24 micrometers for the surface, micrometers squared for the surface area, and 8 micrometers cubed for the volume. That is now a 3:1. And then as we go up and the side becomes 3 micrometers, now we've got a situation where we are 5 to 3. And then that's just going to continue on in that way, with the difference between the surface area and the volume with respect to the ratio becoming less and less. And so the surface area to volume ratio, could look at it almost as a fraction. The surface area to volume ratio is going to decrease as size increases. And that's what you see here. And then this is a graph of this information. This is really similar to the lab you guys did. And this is showing also the decrease in surface area to volume ratio as side length or size increases. Now, cells get around this by saying very tiny, and for larger organisms, instead of just being larger and larger cells, that is where tissues come in, which are comprised of lots of little cells together. So in this case, both of these are ultimately 30 micrometers this way. However, this represents a single cell that size. This is made up of cells with 10 micrometer size. So you're maintaining a lot of membrane this way, and the total surface area is going to be 16,200 micrometers squared compared to, in the case of the single cell, 5,400 micrometers squared. So there's a big difference there. All right, the final thing I want to review in 1.1 is calculation of magnification. Now, I don't have a ruler handy, and you're not going to see this image the exact same size anyway, so I'm just going to guesstimate. But I will show you how this works. So to review, calculating magnification is the magnified size. This is the size that you're seeing the image. So the magnified size, or the image size that you are seeing, and the actual size of the cellular structure or microorganism that you are looking at. Actual size. Okay, so if we're going to calculate magnification in this case, we know that we need the image size, so how large is something in this image, and divided by the actual size. Now, the only information we are given here is the actual size of this scale bar. So that can become our actual size measure, 5 micrometers. Now, for my image size, I can get my ruler, which will have to be imagined in this case. So I'm going to get my ruler, and with my ruler, hypothetically, I measure this to be 2 centimeters, this scale bar. But I need to be in micrometers. So centimeters is 10 to the negative second, and micrometers is 10 to the negative sixth. So that's a difference of 4 decimal spaces. So I'm going to have 2, 1, 2, 3, 4 micrometers. All right, and now I can divide, and 5 goes into 20,000 4,000 times. So 4,000x is my magnification. Now, if I were asked instead to find, let's say there was a line and I'm asked to find the length of this cell this way, well, there's a couple different ways I can do that. I've already calculated my magnification, so I'm going to use this equation here. I know that my magnification is 4,000x, and I don't know my actual size, so I'm going to set that as x. Let's say I measure this, so let me move my ruler from here. And now I'm going to take my ruler here. So let's say I measure that to be 12 centimeters. But I know I'm going to want my actual size in micrometers, so I'm going to go ahead and change that to micrometers. And now I can cross multiply, so the x would go here. And ultimately I'm going to be dividing 120,000 by 4,000. And so now I'm going to divide 120,000 by 4,000, and that is 30. So 30 micrometers would be the length of my cell. And that covers the two things that you're most likely to be asked about, is to calculate magnification or to calculate the length of the actual size of some part of the image, which the scale bar is usually going to be the place you should go in both cases. Calculate magnification, and then you can use magnification to calculate the length of another part of the image. All right, well, that's a pretty comprehensive review of the important things in 1.1. Be sure to also review in your textbook and watch thestudy.com and TED Eds. Let me know if you have any questions.